Good evening everyone. Let's wait for the students to join, then we'll start off with our second marathon. Please let me know whether the audio and the video is clear to everyone or not. Please just confirm. Are you able to see the presentation and is the voice clear or not? Please confirm in the chat sections. Okay, thank you for confirming. Okay, so let's start off with our class. Welcome all to the second marathon series on Botany Insider. We are starting off with the marathon 2.0, and in this again we'll be having 7 classes, 7 topics for you to understand the topics in a much better way and get cleared with all those concepts. Okay, so today we'll be discussing about the nitrogen metabolism. Okay, before starting the video, we are about to start a new course. It is going to be launched very soon on our application, and if you haven't yet downloaded the application, please download it from the iOS and as well as from the Android. Link for the same is in the description box. So if you're targeting the upcoming June 2023 exam, this course would help you out because we'll be having 4 months, like a complete course in which we'll be discussing all the units. We'll be discussing general aptitude, practice sessions, everything would be given to you at one place. Okay, so make sure that you download our application from the Play Store and also from the iOS. And these are the various contents available on our application. We have the 60 classes present. We have general aptitude course, notes booklet, PYQ pointers, etc. etc. And in addition to that, we are about to launch the notes booklet and the PYQ pointers to be delivered right to your doorstep. So we are starting off with the delivery services for our contents so that you can revise the things by the help of the hard copy. So if you wish to have it, you can let us know on our application, on our WhatsApp number that is completely dedicated to the Botany Insider. Okay, this was about it. Let's start off with our topic now. Talking about the nitrogen metabolism. So it is one of the most important topic when you look into the plant physiology. So plant physiology is the 6th unit in the CSIR net syllabus, and nitrogen metabolism is a very important topic. If you look into the previous year questions, if you look into the questions that have been asked, you will see that this topic holds a very good importance. So before starting about the complete nitrogen metabolism, we need to have an understanding about like why is nitrogen very important, because if we are discussing about the nitrogen metabolism, so जो nitrogen है, ये जो element है, what is its main role? Why is it so important for the plant? So nitrogen, it is an essential element that is required in the greatest amount. और अगर हम environment की बात करें, so 78% of the atmospheric air, if you look into here, 78% of the complete or of the total proportion of the air present, we have 78% oxygen, nitrogen, we have 21% oxygen and 1% other gases. Next, in proteins, when you look into the protein structure, nitrogen makes about 10 to 12% of the cell. They are the building blocks of the amino acids which are synthesized when the inorganic nitrogen converts to the organic nitrogen. That means, as you all know, that amino acids are what? They are the building blocks of proteins, right? And in the building blocks of protein as well, these amino acids are synthesized when the inorganic nitrogen gets converted into the organic nitrogen, and they play a very important role in growth, reproduction and the heredity. Talking about the source to the plant, so from here the previous year question have been asked. So it's a very important thing to remember that atmospheric nitrogen is the molecular nitrogen, then nitrate, nitrite and ammonia in the soil is there. Nitrate is the chief form, then we have the amino acid, that is the organic nitrogen in the soil, and the organic nitrogenous compounds in the body of the insect. So all these are the sources to the plants. Talking about its constituents, so nitrogen is found in chlorophyll, cytochromes, alkaloids, vitamins and the nucleic acids. So till here is everything clear to everyone? This is just the introduction for that matter. If there's any doubt, any problem, please ask, otherwise we'll move forward. Okay, any doubt, anyone till here? Yes, quickly. Then we have the main nitrogen cycle, that is the main concept of our nitrogen cycle. Okay, so we have the nitrogen in the atmosphere which is in the form of N2. Then this N2 needs to be fixed. So there occurs nitrogen fixation. Now this nitrogen fixation can occur in the bacteria and the plant roots and the soil bacteria. So both of them converts, first process is the ammonification. Now this ammonium, that is NH4 plus, gets converted into nitrites via the process of nitrification, and these nitrifying bacteria convert these nitrites into the nitrates, that is NO3 minus. Now what happens, these nitrates, they get assimilated, that is they are used up by the plants are eaten up by the animals, and as and when the plants and animals die, we have the nitrogen containing organic compounds which after the death decomposes and converts back into the NH4 plus. In addition, these NO3 minus, they are like via the process of denitrification, we have our final again atmosphere in the nitrogen. So this is how the complete cycle, or this is how the complete nitrogen cycle actually operates. All of these color codes would help you to remember about the main processes. Main thing is the assimilation, ammonification and the nitrification, and giving the nitrogen back to the main atmosphere, we have the denitrification all and over again. So this is about the nitrogen cycle. I hope this thing is clear. Very, very important. Time and again questions get asked from this particular area, that is the various types of the steps that gets operated. In addition, you can remember about the examples of them as well. So if you look into here, so first we have ammonification, then we have nitrification, and we also have the denitrification. In the ammonification, what do we have? We have the organic nitrogen, that is the N2, via the help of ammonifying bacteria or the ammonifying organisms, they get converted into the ammonia, that is NH4 plus, and conversion of the organic nitrogen into ammonia is via the, or it is a reductive process. All the things written on the screen are very, very important. Please make sure that you remember about them. Then after this, what do we have? We have the nitrification. So nitrification, main thing is the NH4 plus, that is the ammonia gets, or the ammonium ion gets converted into NO. So this is the oxidation process. Ammonium ions gets converted into nitrite, and nitrite get converted into the nitrates. Yeah. They gets converted into the nitrates. So NH4 plus into NO2 minus, that is NO2 minus, it is via the nitrosomas, and nitrite into nitrate is via the nitrobatter. Okay, last we have the nitrification, denitrification. In this what happens, we have the NO3 minus converting into NO2 minus, then NO, NO2, and finally the main dinitrogen. So the denitrification occurs by the help of the denitrifying bacteria. We have main two examples. First one is Pseudomonas and the second one is Paracoccus denitrificans. So I hope all these points are clear, because time and again questions are asked from here. If you have any doubt, any problem, you can ask. Otherwise we'll move forward. Okay, any doubt till here? These are the main steps of three main steps of the nitrogen cycle which you need to remember. If you have any doubt, any problem, please ask. Otherwise we'll switch over to our next slide. Very important concept. Time and again questions are asked in the previous years, so in the subsequent years as well you can have questions from this area. Clear? Okay, do we have only one student replying for all the questions? Why is not anyone else replying? Like, are you not interested in the class or what? We have 7 students watching the lecture. Why is only Farheen replying to all the answers? Are you not able to understand others, or are you just sitting here for formality? Let's move forward and understand about the biological nitrogen fixation. So in this, what actually happens, we have the conversion of the molecular nitrogen into the molecular nitrogen of the atmosphere into the inorganic nitrogenous compounds. And these inorganic nitrogenous compounds, when the nitrogen fixation occurs via the help of the living organisms, it is given the name of biological nitrogen fixation. So in the nature there occurs nitrogen fixation via 3 processes. First one could be the biological one, that is 90%. 8% of the nitrogen fixation occurs via lightning, that is via the natural process, and 2% occurs via the help of the UV radiations, that is 2%. So remember about this thing as well, very very important. I hope these basics are clear, because if the base would get cleared, understanding the other things becomes easier. So now we have the nitrogen fixing organisms. So in the nature we have various type of organisms present, right? So only some of them have the capacity to actually fix the nitrogen. We have either the free living or the symbiotic. In the free living we have autotrophic and the heterotrophic, and in the symbiotic we have the root nodule concept. So in the free living autotrophic, in autotrophic as well you have either the aerobic or anaerobic. Aerobic example is nostoc and anaerobic example is rhodospirillum. All these examples are very very important. Please remember about them. Then we have heterotrophic. In heterotrophic it could either be aerobic, facultative, or the anaerobic. Aerobic is azotobacter, facultative is bacillus, and anaerobic is clostridium. Last is the root nodules. In the root nodules it is actinorhizal and others. So please remember each of these examples. All these examples are very very important. Every single point written on the slide is of importance. Like, you can find questions at every single point, so please remember about them. Then let's look into the main mechanism of the nitrogen fixation. First thing first, this complete fixation occurs via the symbiotic association. Symbiotic association of what? Of the bacteria that is working, and in addition we have our main plant as well, right? So what is the plant giving to the bacteria? It is giving the sugars. And what is the bacteria giving to the plant in return, it is the fixed nitrogen. So that is the reason why it is symbiotic, that is both of the members, that is the plant and the bacteria, they both are positively helping out each other in the process. Then to the host, bacteria provides the main fixed nitrogen, and to the bacteria the host gives what? It gives the carbohydrates and the shelter. So carbohydrate is the succinate, malate and the fumarate. So please remember about this point as well. I guess in 2015 a question was asked related to the same concept. So please remember about it. Talking about the requirements of the complete fixation. So there are 4 main requirements. First one is the nitrogenase enzyme that is present in the nitrogen fixing organism. We require energy, that is the ATP. Proton is required, and finally we have the ferredoxin required in the complete process. Now this nitrogenase is an enzyme complex which contains 2 subunits, which contains 2 metalloproteins. Which are those? First one is iron proteins and the second one is iron molybdenum protein. So this iron protein is what? It is the azoferredoxin, which is smaller, like both of these are the subunits, right? These are the subunits of the complete nitrogen molecule or we can say the nitrogenase molecule. The first subunit is iron protein, which is the which is given the name of azoferredoxin, which is smaller in size, sensitive to oxygen, and it is a dimer having 4 ions and 4 sulfurs. Then we have FeMO protein, which is molybdenoferredoxin, which is a larger subunit having 2 different peptides, which is 2 alpha and 2 beta. So it is a tetramer. Please remember all of these basic details. These are very, very important, and every single point written on the slide is of utmost importance, because in 2019 question was asked from this area. In 2016 and 15 both question was asked from the carbohydrates. In 2013, if I'm not wrong, requirements were asked, like in a statement wise question this particular point was given, and one more point was there, that is nitrogenase is present in the nitrogen living organism, that is the nitrogen fixing organism and not the main plant. So remember this thing as well. Then one more important point was asked regarding the biological nitrogen fixation requires whether the anaerobic or the aerobic condition. So it requires the anaerobic condition, that means it requires absence of oxygen. Why does it require absence of oxygen? Because the nitrogenase enzyme, which is the main enzyme in the complete functioning, it is oxygen sensitive. So if it is oxygen sensitive, it cannot work properly in the condition of oxygen, in the presence of oxygen. So that is the reason why nitrogenase, as nitrogenase can work only in the absence of oxygen. So that is the reason why this complete biological nitrogen fixation occurs in the anaerobic condition. Okay, is this topic clear to everyone? Yes or no? Yes, is everything clear till here? Any doubt? Yes, any doubt? Very good. Let's move forward now. Now talking about the complete equation or the complete process of the nitrogen fixation. So we have N2, 8H plus, 8 electrons and 16 ATPs working in the complete process. It leads to the formation of 2 HNO3 plus H2 plus 16 ADPs plus 16 PIs. It got, the orientation got disturbed, so it is 16 PIs. So there was a question asked related to the number of ATP as well. So there are in total 16 ATPs that are required in the complete biological nitrogen fixation. Okay, please remember all these points. All these points are very, very important. Okay, I hope this point is clear. In addition to that, hydrogenase enzyme is also found in some of the rhizobias. Why is it helpful? It helps to split the H2 into 2 electrons and 2 H plus. As you can see that H plus is required and electrons are required. So both of these things get fulfilled via the help of hydrogenase enzyme as well, and this leads to increase in the efficiency of nitrogen fixation. So because the nitrogen fixation require both H plus and electrons, and this hydrogenase is helping in the splitting of H2, so we can say that the efficiency of nitrogen fixation is getting increased. Okay, so I hope this thing is again clear to you. Let's talk about the genes involved in the complete process. So the genes involved in the so the genes involved in the nodule formation are, we have two main genes. Again, previous year questions from here as well, question have been asked previously. So talking about the genes involved, so there are two main genes involved. First one is the host specific, second one is the rhizobial specific. Both of them are given the name of node itself, but host specific is capital NOD, which is the nodulin gene, and the rhizobial specific genes are small NOD genes, that is the nodulation. So remember it by the help of this. Now these genes, that is these nod genes, are again of two types. That is, first we have the common nod genes and second one is the host specific. So common nod genes are which one? These are nod A, nod B and nod C, and host specific are all the leave ABC, other ones, that is P, Q, E, F, D and DL. All these are the host specific. Again, question have been asked like 4 to 5 times from this area as well. Remember about the common nod genes being ABC. Except for ABC, all the other ones are host specific. Why are they actually needed? They are needed for the synthesis of our nodulation factor, so that the rhizobial specific genes could get activated and the process could initiate. Then talking about the nod factors, which are the ones in the rhizobia or our nitrogen fixing organism, these are, or they are having the nature of lipochitino oligosaccharide. They have their products. So because in the nod genes we have ABC, nod A, nod B, nod C, each of them have a specific product. So nod 1 has product of capital NOD A. So what does this mean? As and when the rhizobial genes gets activated, they lead to the activation of the genes in our plant or in our host. So capital NOD A is N-acetyl transferase. Small NOD leads to the activation of capital NOD, that is chitin oligosaccharide deacetylase, and third one is NODC, that is not C, chitin oligosaccharide synthase. Please remember about this thing as well, very, very important. Okay, let's move forward and understand about the formation of the root nodules in the leguminous plants. If till here, if there's any problem, please ask, otherwise we'll move forward quickly. Any doubt? Yes. Okay. Let's move forward now. Let's talk about the formation of the root nodules in the leguminous plants. So talking about the rhizobium, which is the main nitrogen fixing organism. It is free living before the infection. It is a gram negative bacteria, motile in nature, rod shaped. It is a proteobacteria. Main genes, the genes that would get involved are small NOD genes, and which is we have a Rick Addison, which is present on the surface of, or which is present on the surface, which plays the most important role in the plant bacterium attachment. That is, you have the plant, you have the bacterium. So how these two gets attached together in order to initiate the infection? This occurs via the help of the Rick Addison. And where it is actually present? It is present on the microbe. Which one? That is the rhizobium. Okay, easy to think to remember. Talking about the nod genes, we have already discussed about these. What is the main role of it? It helps to induce the root hair curling, which triggers the plant cell division, and as a result of the same, it leads to the initiation of the complete nodule formation. Okay, then we have the various steps to the root nodule formation. There are majorly 6 steps for the same. Look into the figure, you will understand. First, what will happen? The root hairs would be releasing a chemical signal which leads to the attraction between the rhizobia and the plant. Then curling of the root hairs would be done, leading to the formation of infection thread. Finally, the infection thread would grow to an area where the number of rhizobias could grow, and finally we have the formation of a nodule like structure where our rhizobia could stay and it could grow. So first we have the rhizobia, free living organism before the complete process, before the complete signal gets exchanged. Then there occurs exchange of signal, infection thread gets formed, cortical thread is there occurs enlargement, and finally a group of bacteria over here that is given the name of bacterioids. So we have a nodule like structure formed, that is this kind of structure. So we have various bacterioids present, and the surrounding membrane is given the name of peribacterioid membrane. So this was about it. I hope this thing is clear. Very, very important. Look into the figure, look into the steps, you will understand. I have tried to make this as simple as possible for you all. If you have any problem, please ask. Yes, very, very simple. I have tried to make it very simple for you all. I hope this thing is clear. Let's move ahead. Now, what are the steps? We have the attachment. The membrane gets dissolved. It enters the root here. Infection thread gets formed, and finally the root nodule is formed. So when the infection thread is there, so first the bacteria is in the roots. It reaches the cortex, forms a membrane around itself given the name of peribacterioid membrane, and finally this kind of structure is formed. Then the root nodules of the leguminous plants is the color of the root nodules is red color. Okay, peribacterioid is the outer membrane into which the final or the complete nodule is there. So it is like a, what do you say, a bubble like a structure where all the bacteria are surviving. Difference between cortical and infection thread. Cortical is a region in the complete area. So if you remember about, just wait a second. Yeah. So cortex is the region in the root. Okay. Infection thread is the thread via which the rhizobium or the microbe would be entering from the outer environment into the root area. Okay. So this is the difference between the two. Next question was, Ayush, we have a marathon running for the time being. So for the next 7 days, we'll be having a daily class. We have our own application as well. You can join in there. It would. And you can enroll in our live courses where we have a structured, like complete structured courses running for the complete preparation thing. Okay. So Fareen and Anoop, is your doubt clear or not? Peribacterioid is the outer membrane, cortex is the region, and infection thread is the thread, or it is the main pathway via which the microbe would be entering from the outer environment or from the soil into the root region of the complete plant. Clear? Any other doubt? Okay, let's move forward now. Last point, that is this one. Okay, then so the root nodule of the leguminous plant is reddish or pinkish in color. Why is it? Because of the presence of a red pigment given the name of leg hemoglobin. Then the oxygen binding heme protein occurs in the cytosol. All these points are very important. Please remember, it gives greenish pinkish red color to the nodules. Parts are like, when you look into the hemoglobin part, so we have the heme part and the globin part. So the heme part of the leg hemoglobin, it is synthesized by the bacteria, and the globin part is synthesized by the host. So as in when the host and the bacteria, both of them interact with each other, it leads to the formation of leg hemoglobin, and finally the nodulin gives a pinkish appearance, or we can say a reddish pink appearance. This question has been asked in the previous years in the GATE exam, in the DBT exams as well, so you can remember about it. So now talking about the last point related to the factors affecting or factors controlling the nitrogen fixation. First is the environmental factors, then we have the stages of plant growth, fertilizers in the soil, and the genetic factors. So environmental factors like temperature, adequate moisture, sunlight, and carbon dioxide levels, all of these help in the initiation or helps in the, what do we say, encouragement for the nitrogen fixation. Then talking about the flowering season. So in the flowering season there is more of biological nitrogen fixation. Fertilizers lead to increase in the fixation, and the genetic factors, that is genes, also play a very important role in the complete nitrogen fixation. So these were the main points, these are the main important things that you need to keep in mind when you are studying about the biological nitrogen fixation. So if you all have any doubt at any point, please ask. Otherwise, this was about the complete nitrogen fixation from my end. If you have any doubt, please ask. Otherwise, we'll complete the class. Clear? Okay, Harshit, others? Any doubt? In addition, we have a special 25% flat discount running on our application on the occasion of Christmas and New Year. If you wish to have any course, any of the content, please have it from the app. A flat 25% off sale is going on. Get your content in the lowest price possible and strengthen your preparation. Okay? You're most welcome, Anoop. If you wish to have any kind of content from our end, download our application and get the notes, get all the PYQ pointers and various courses, live courses running for the same with 25% off. And we're about to launch a new course for the June 2023 paper. So if you wish, you can enroll in that. It starts very soon. It would be a very structured course containing all the units, all the previous year question sessions, every single thing in the same course in minimum price. Enroll in that. It would be launched very, very soon. So this was about for today's session. Thank you for joining in. These are the contents on our application. Currently we have 25% flat discount running. Enroll in these courses with the help of the same. You can have the presentations as well. You can have the live sessions, PYQ pointers, general aptitude, etc., etc. So all these things would help you to strengthen out your preparation. Thank you for watching this video. Thank you for joining in. Let's meet tomorrow. Tomorrow also we'll have a new topic discussed in a full-fledged manner in the same way. Let's meet tomorrow. Bye everyone. Take care.